64.
All right, folks, I see a lot of you are starting to come in. We're going to get started in just a little bit. I'll let the room fill up first. Good evening, everybody. A lot more people still coming in, so we'll just give a few more minutes. And I say good evening because we are mostly based in New York for the meetup is, but I know we're pulling people from all around the world. So if I say good morning or good afternoon or good middle of the night, I hope everyone's covered there. We're very excited to do our first ever completely remote meetup. It is not actually our first ever remote meetup. One time, maybe 10 years ago, we did have a meetup when the bulk of the people were all physically in New York, I believe at the AOL offices, and we had someone remoting in, I want to say Australia, but I could be making that up. I know someone actually had to do a video, one of the presenters actually did it via video, and this was 10 years ago, so it was very exciting for us. I'll give everyone just a little bit more time. Then we'll get going just a little bit. Hope everyone's having a wonderful, wonderful quarantine and making the most of it. Hopefully everyone's doing well and is healthy and their families are all in good situations. And hope those of you with kids are surviving and getting through it. And those of you without are writing a great R package. And I hope everyone's just doing the best they can during these times. Okay. Yes, so some people, it's just on the Q&A. Thank you for letting us know, Avik, about the password. We made a last minute decision because we realized that some people are having trouble getting the password. We turned off the password because it's in webinar mode. So sorry everyone for that rush, but since it's in webinar mode, no one should be able to show any video or make any audio unless we turn it on for them. So we felt safe for this instance, removing the password at the last minute. Hopefully there won't be any issues, and next month we'll see how we'll do it then. We might not even need to have a password in the first place. So thank you for pointing that out, Avik. All right, it seems like we have the number of joining us stabilized. So first up, everyone, welcome to the April 2020 meetup. Hope everyone's here is ready to learn a lot about R, a lot about coronavirus. We have an expert in both of those leading us today. So first thing, we always start up the meetup with who's hiring. And since we are in webinar mode, we can't have everyone raise their hand and say who's hiring. So if you could all go to the Slack team that we have for NYHackR, there is a job postings channel. So if you need to find that, I will post it in the chat here. nyhackr.org is the meetup's website. And if you go there, you'll see there's a page called Slack. If you're on mobile, you might need to scroll to find it. There's a Slack page, and then there's a link for that. Amada, actually, could you post the link directly to Slack from the page? That'll probably be easier. And if everyone would like to join the Slack team, there is a new channel for the meetup tonight. Amada, what's the name of the channel you created?
It's April 20 -meetup.
April 20 -meetup, that is the name of the Slack channel for today. If anyone wants to jump in there and chat, I figure that's probably a better place than the random channel or the general channel. But if you're on there, you want to post a job. If you're hiring, there is a job postings channel. Go into job postings, post your job. If you have a link, post a link for it, say who the job is, where the job is at, who it's for, who you're looking for, and hopefully you'll get some good responses on that. So again, that's in the NYHackR Slack. There is a jobs channel. And just a reminder, Amada created the April 20 meetup channel. So go there and chat away about the meetup, about tonight's meetup. We'll be looking for questions. Oh, hello, Stephen. Thank you for joining from Boston. So if you have questions, you can ask them in either the meetup channel or here in the Q&A, whatever you're more comfortable with. And we will go through them and try to pitch them towards Noam at the end of his talk. So, save your questions toward the end and we will go through them and ask those questions to Noam. If you're interested in getting more involved with COVID efforts, there's also a COVID-19 channel where a number of people, including Noam and Amanda, and I think Rami is in there. These are all first names, I understand. But in there, you'll find resources for going and trying to help people with COVID. I just realized my video turned off. Sorry about that, folks. So if you look on the Slack, there is a COVID-19 channel. And in there, you'll be able to share information about COVID, different things you want to do, different tools you're using, or questions you have. If you just want to ask questions because you don't understand, we have a few people who are experts in coronavirus in there, namely Amanda, Noam, and Rami. I'm sure others, but those are the three I know who can help answer your questions about COVID in general, about R and COVID. Normally at this point in my announcements, I say there's a pizza poll and we ask questions about pizza. And so unfortunately we can't all be together, but I did make sure to go out and get some pizza. You can see it is exhibiting good cheese lock. The cheese is not sliding off. It's a little not charred too much on the backside, so that's not great, but not a lot of tip sag, just a little, which is what you want. So hope everyone has their own slice of pizza and is enjoying their meal as Noam speaks. I'm sorry we can't all be together, but this is the best we can do in these times, and hopefully everyone can enjoy meetup in the comfort of, enjoy the meetup and their pizza in the comfort of their home. Ordinarily, we like to go to the bar afterwards. Unfortunately, we all can't go to the bar, so everyone can hang out on the Slack channel and talk virtually, or if there's real demand, we maybe could spin up another Zoom, maybe. We'll see if it's in our capabilities. Everyone can grab a beverage, alcoholic, non-alcoholic of their choice, and hang out if they would like to. Next month's meetup, we think we settled on a date. I just need to confirm with the speaker, is this gentleman Rami I keep mentioning. He built the coronavirus package in R amongst some other resources. So we're assuming we'll be remote again next month, and we will do another remote meetup, and Rami will have another talk about coronavirus because I have a feeling people will still be interested in it in May. So please, everyone, please look out for the announcement in the next few days for the next meetup. There is a STAN workshop this week, Wednesday through Friday. It's all remote. I'm going to be doing it through Zoom with passwords and webinar mode and all that. If anyone still wants to sign up, you can still sign up today. I think we're closing registrations today just to make the logistics all a bit easier. But that will be, sorry about that, I dropped my phone. That will be this week. Jonah Gabri, Rob Trangucci, and Ben Bales will be leading it, and Andrew Gilman will be popping by to help out. And that'll just be a great time. It's our annual STAN workshop, a great way to learn about STAN and Bayesian data analysis. You can find more information at nyhackr.org. I'm sure a lot of you are interested in the New York R conference. This year is supposed to be our 6th annual R conference. Right now it is scheduled for June 11th through 13th. We are deciding later this month if we have to postpone it to later in the year or if we have to go all virtual. So check that out at rstats.ai and we can figure out more there. We will post more information on rstats.ai over the next few weeks if we decide to reschedule it or go virtual, or even if we're lucky, go as planned, then we'll see how that goes. We'll follow governmental advice. I see someone's asking a question about the Slack link maybe not working. So you might want, that might be the old link. So if you go to nyhackr.org/slack.html and click the link on that page, then that should take you to the fresh link, and the model will try to grab that and paste it. We had to renew the link recently, so hopefully the one she just put up there will work this time. If that, that's app.slack. Sorry folks, let me just go try that. I don't think that's the right one. Let me go grab the link that's up there and people could tell us if it works. Because that looks different than this one. Try the one that just says join.slack.com and see if that works for you folks. Hopefully that'll be hopefully that'll fix this. So I mentioned NYR. We also have Washington DCR. That's scheduled for November. So as of right now, that's going ahead as scheduled, and we'll see what happens with the virus. So that again needs to be rescheduled or go virtual or whatever it may be. So with all that, I hope everyone remembers, post your jobs on the NYHackR Slack channel for job postings, chat about today's meetup in the April 20 meetup channel. Enjoy your pizza at home if you can. And think about the STAN workshop this week. And next month, again, Rami will be giving a talk about coronavirus, most likely remote. We are pretty certain it's going to be a remote meetup. Well, especially since he's from California, so it's definitely going to be a remote meetup, even if we can go in person at that point. So with that, I would like to introduce our speaker. He's spoken a few times at the meetup. He works for an organization called EcoHealth Alliance, which specializes in the study of emerging viruses. And in particular, Noam knows quite a bit about coronavirus. And you'll see on the meetup page is a picture of him holding, it's a form of a rodent. It might be a pangolin. I'll let him tell you what that picture is. Him holding a pangolin in a rural part of China because he studied this. So I couldn't think of anyone better to give a talk about coronavirus NR than Noam. In addition to what the normal talks he gives about GAMs and Docker images, I figured this is perfect for him. So everyone, please welcome Noam Ross.
Thank you very much, Jared. And thank you all for coming today. We have over 200 people. This is way bigger than a typical meetup that we have for New York R. I racked my mind a lot about what to talk about for this meetup. We actually hatched the idea originally back in, I think it was mid-January. Jared said, hey, do you want to give a talk in the spring? And I said, you know, I've talked about workflow and I could talk about Docker and I could talk about certain types of models, but, you know, there's this thing happening in China that's in the news that people might be interested in. I actually can talk about the substantive research that I do. And Jared said, well, I hope it's still newsworthy in April. And unfortunately it is. And I've wrapped my mind a bit about what to talk about for this, because, you know, this is our meetup. Many of you aren't part of the regular meetup group that we have, but the New York R community is a tight one and we have a lot of fun, and it's a very somber topic these days. For those of us everywhere in the world, this has profoundly affected, you know, our lives. For those of us, especially in the hot spots, it's been a scary time and a somber time. You know, and, you know, there's a lot of suffering out there because of this. And I thought, well, what can I talk about? What do people want to hear? And so I thought this is still our, you know, our meetup. This is still our meetup of people who are interested in models and statistics and problems and interesting data. And I'm not so much going to try to keep it lighthearted, but try to, you know, spark and sate some curiosity very much on the, you know, on the modeling side to talk about how and where we use models. A bit about myself, as Jared said, I work for a nonprofit called EcoHealth Alliance that's based in New York and works at the intersection of wildlife conservation and human health. So we very much focus on emerging infectious diseases. I'm a disease ecologist, and what that means is it's like disease ecologist is to epidemiologist as veterinarian is to doctor. So I study the circulation and emergence and dynamics of diseases predominantly in wildlife and how they emerge into humans. And so it's very much the same set of tools that infectious disease epidemiologists use. And for those of us who work in the space where those things interact, we work very closely together. I also work for an organization called rOpenSci, which is a developer collective to support technical and community infrastructure for R to support open, reproducible science. We like to say that we build communities as much as we build packages. rOpenSci is an open volunteer organization that helps support scientists do their work and get access to open data and create open data. So what I thought I'd talk about today is how do models and how does software help us understand emerging diseases like COVID-19, like other diseases like SARS and Ebola. And like I said, what I wanted to do was focus not just on coronavirus, but sort of sate people's curiosity and pique their interest into the types of models that we use to understand things. So many people here are data scientists of various sorts. They have modeling workflows and tools that they use. And what I wanted to pose was several scientific questions that I and other disease ecologists and epidemiologists work on. And with those scientific questions, ask, well, what are we trying to find out? What models do we use to help answer those questions? And then what tools do we use for those models? Because maybe some of you will be able to pick up those tools and take them and use them in your work, be it COVID or disease or health related or not. But maybe some of you will learn about the methods used in those tools and be able to apply them to other things. Hopefully it'll help you all understand something about the news and the research and everything that comes out that starts talking about models of infection disease. It's a high point in the culture for infectious disease modeling. Things are in the news that have really rarely been in the news before, and we're inundated with a lot of information, and being able to look at that and critique it and think about what those models actually are so that you can compare them to the other types of models that you may be using in your own work and apply your own data science and statistical backgrounds to understanding them is something that I hope you'd get out of this talk as well. So I'm going to speak about 3 questions, and then afterwards I'm going to talk a little bit about the types of things that people who are on this webinar or part of the meetup or elsewhere can do to apply their skills to problems related to coronavirus. So I'm going to talk about 3 types of scientific questions that we use models to answer. The first one is, how do we trace the source of a disease? When we discover new pathogens, new viruses, how do we figure out where they come from? The second question is, where are the sources of new diseases, future diseases? How do we understand where risk is and what to focus on and where to do surveillance and monitoring to understand the potential for future outbreaks? And then I'm going to talk a little bit about how we understand how diseases spread in populations. How do epidemics grow? What stops them? What can be used to forecast the future? And for each of these, I'm going to talk a little bit about what the problem is, what the nature of the data and the models are we use to solve, and then go a bit into the R packages we use for those. So the first of these is, how can we trace the source of a disease? The picture I'm showing in this slide is in a cave in Yunnan province that is full of horseshoe bats, and a couple of people from my organization there who work in China sampling bats to discover the viruses that live in them. Many of you have probably heard in the news that COVID-19, which is caused by the coronavirus now called SARS-CoV-2, has its origins or is derived from or is evolved from bats. And how do we know that? And how do we learn that? And how do you use data and models to determine that? Well, it starts with the data. First, a lot of data has to be collected in order to compare things to that virus. And that happens via people doing the type of work like they're doing in this picture, which is sampling both wildlife and people who work with or near or live near wildlife, sampling things like blood samples or saliva samples or fecal samples, and doing testing to determine what viruses are out there, what viruses are circulating in the general population of different types of animals, or maybe occasionally spilling over into humans, that potentially could cause outbreaks. And what that does is that creates a bank of data against which new viruses can be compared to. So let's think about the type of data one has when you have a new virus and you need to compare it and try to understand its source. So this is the type of data that one is working with when one's trying to understand the origin of viruses. It's genetic data. So genetic data comes from genetic testing and sequencing. And when one gets a sample from a wildlife or a human, you can identify the viruses in them, collect the DNA or the RNA in those viruses, and use molecular processes to sequence those, and you get a sequence of letters or nucleotides that make up the DNA or RNA sequences of these viruses. And they look like this. It's a string of letters A, T, G, and C. If it's RNA, there's a U in there as a replacement, which is a different molecule. But this is the genetic sequence of viruses, and this is the type of data one is working in. One might have a new virus, and you might want to compare it to that bank of other viruses that have been collected by wildlife researchers and virologists over years and decades, and banking them in these genetic data archives. One note is that the sequences themselves are models. They're not just data. Like everything, the data collection process is noisy, and the chemical processes that one uses to extract these RNA and then try to align segments of them together and determine how much noise there is over reading them over and over again introduce errors. So these sequences themselves are sort of consensus models of doing that sequencing over and over again. But it is something that's standardized enough, and we understand those models well enough that you can have pretty reliable consensus sequencing, although there are certainly cases where that type of error is important as well. So if you were a data scientist and not necessarily someone who worked in virus or genetics, and you want to understand the relationships between, say, this new virus's sequences and these genetic sequences from a series of other viruses that might have been collected, and you want to understand which ones are closer to each other, you might use something like some sort of clustering algorithm approach. You might use something, say, like string distance, or you can use the string dist package and use any number of functions that measure the difference in overall substitutions between two strings, and you could come up with some sort of relationship of the difference between different genetic sequences. And this is exactly sort of one of the approaches one can use in order to compare genetic sequences. And in the genetic world, a common algorithm that's a clustering algorithm used for this is what's called generating a median joining network. So the median joining network is the medium best consensus network joined by just a straightforward difference clustering algorithm. And the picture I'm showing here is one of these median joining networks that compares viruses that were collected from individuals who had SARS-CoV-2 with viruses that were in any number of species of bats in Southeast China, South and Southeast China, that had other coronaviruses. And so this median joining network gives you an idea of the relationships amongst those viruses. And this is sort of a straightforward clustering algorithm that's used as sort of a first pass of understanding relationships amongst genetic sequences. It's a sort of fast, quick, and dirty way of doing this that gives you an understanding of the topology of a network like this that requires very few resources and lets you understand how to pursue your data analysis from then on. So you can have this clustering approach. But when you really want to look more carefully at the relationships amongst genetic sequences, you want to introduce more knowledge into the process. This is sort of a naive clustering approach, but we know there's a lot of biology and process and nonlinearity in how genetic sequences relate to each other, because they're not just sequences of strings and differences. They're the result of evolution, and the evolution process is directed and makes certain patterns appear in genetic sequences that one has to account for. So usually when you see how organisms are related to each other, you don't see it by this sort of undirected network approach, but instead you see something that generates something called a phylogeny or a phylogenetic tree. And that's the kind of thing one uses in order to understand the relationship of viruses or other organisms to other organisms that might have common ancestors that they evolved from. So this figure that I'm showing on this slide is from a paper that characterized the SARS-CoV-2 virus, which causes the COVID-19 disease, that was published just a couple months ago in February, really rapidly, really fast turnaround in understanding a new virus like this. And what they did here is they generated this phylogenetic tree, which is another network representation of this genetic information, but one that explicitly includes the evolutionary structure of those relationships. So in red, we have in the bottom red, a bat virus that was collected in southern China several years ago. And then several other samples of the, what at the time was just novel coronavirus and now is SARS-CoV-2. And by explicitly using sort of evolution as part of the structure of one's model, you can create these phylogenies. So the phylogeny itself, this tree, it's not data. The tree itself is a model. In this case, you make what's called a maximum likelihood tree that uses a maximum likelihood approach, much like one does in, regression models. And trees like this also have error and uncertainty. And so one often uses a bootstrap approach in developing trees like this. If you can see, the branches of these trees have little numbers on them, and that's the fraction of the percentage of the bootstrap that's the percentage of the bootstrap samples that support that topology. So there can be uncertainty in these trees. And if you're looking at something like this, you see it in a paper, you see it in the news, you see someone recreate it. One of the important things is to understand the nature of the uncertainty in that topology, because a tree is not an absolute thing any more than a regression line is. It's a model that's created out of data. And when one's building things like these phylogenetic trees, you have a whole lot of different approaches and a lot of considerations, and you have many of the same challenges that one has when doing other identifying, other model building, other machine learning. So one might take a clustering, a non-parametric approach that just links things together. You can do this maximum likelihood and bootstrapping approach, in which you try to find this tree that best represents the evolutionary process. And you can go all the way to Bayesian inference. So, just like in a regression model, you can do a maximum likelihood and confidence intervals, you can represent a whole posterior. You can get a whole posterior of different trees, and you can interrogate that output and understand that posterior and ask questions about things that are uncertain and you're not uncertain. And when you're doing that, these trees often aren't simple because the evolutionary process is not simple in itself. You have to think a lot about a lot of things like the mutation rate that's actually going on in the genes. That mutation rate might change in time, or it might change along different parts of the sequence because different genes have different mutation rates, perhaps because some of them represent something which is under evolutionary selection, and some of them may not. Also, while in mutation, you just think of like individual genes or individual proteins changing bit by bit over time. Sometimes you have other things like recombination, which reshuffles the genetic material and it really would throw a wrench into your phylogenetic tree process if you couldn't explicitly account for it somehow. So just like when you're doing a regression model, you have to start maybe thinking about, oh, perhaps there's time or space and autocorrelation has to be included, or maybe this component should be a random effect or fixed effect. These types of considerations go into the process of generating phylogenetic tree models, and you have to tweak them and identify the right sources of heterogeneity that you need to explicitly include in the model, maybe because you need to correct for it and you're trying to control, or maybe because it's the thing you're most interested in. Because understanding like those variation in mutation rates helps you, for instance, understand if parts of the genetic sequence are evolving. So just like how in R, there are 100 packages for every type of regression, there's a whole world of phylogenetic modeling software for making these trees and for taking different approaches. So there are a couple called, there's RAXML and IQ tree, and those are maximum likelihood approaches. The first one is a rapid algorithm for ML. And then there are a couple of software packages that are for Bayesian evolutionary analysis for generating all those posteriors of trees. Much like a lot of other software in R, these things often have to be coded in low-level languages like C in order to get the performance you need. But that doesn't stop our users. If there is a useful piece of software out there and our user will write a package in order to connect to it and use it and create bindings to use things. So there are things like the IPS package that is the interface to phylogenetic software, or the HarrietR for IQ tree. And there's a fantastic package that I like to promote called BeautyR for Beast. One nice thing about that is it's an rOpenSci peer-reviewed whole suite of packages for people who use that type of tools. So like some of these other ones, it's been validated for through a peer review process, not just of the approach, but the code. And that's one of the things that rOpenSci does that helps scientists. And so you get this whole world of software, and it's sort of like probably parallel to the things you're used to using for regression or Bayesian analysis. And when you see trees that represent the relationship between viruses or dinosaurs or anything like that, you can understand that you have the same type of processes going on in trying to develop those and do inference on those and use those to understand the nature of the evolution and the origin of species. There's one application of this relating to COVID that I really like to highlight, which is a website called nextstrain.org. Nextstrain was originally created to track flu-related evolution and has been used to track all sorts of viral outbreaks worldwide, and is a tremendous effort out of the University of Washington, but with a global team and lots of participants to continually track the sequences of new viruses as they are collected worldwide. And there are more than 10,000 samples, I think, of COVID virus genes that have been posted to a public site called GISAID, and that Nextstrain has used to create this mass tree. And this really helps understand not just the origin of, say, the virus to its wildlife host, but the origin of clusters of viruses and infections, so you can understand something about how the disease spreads in time and space, because one can track clusters or strains, or what they call clades, of the virus to the set of viruses that they were imported from. And that helps researchers understand movement in time and space. So if you go to nextstrain.org, you get these fantastic interactive trees that are linked with maps. And they show how this sequence had an ancestor related to another sequence. For instance, it was something that was used at the first outbreak in Seattle, or outside Seattle in Washington, to show that there was community transmission because different sequences were clearly related to each other and not being imported from separate places. So one could distinguish community transmission from new imports by looking at the phylogenetic trees of those genetic sequences. One of the things we've learned in New York is that many of the early cases originated from Europe, and we understood that from comparing European sequences to the sequences from early cases in New York. And there were lots of those sequences relative to, say, sequences from Asia. So this is something that is interesting, but is also an important epidemiological tool to understanding and reconstructing the spread of an outbreak. So I'll jump to a different question here, which is, what are the sources of future diseases? So I want to again, return to this photo about of the collection of viruses from bats in this cave in China, because it's not coincidence that people were there researching. It was part of a large-scale project to collect viruses and understand the distribution of viruses globally and the potential for outbreaks. This one was under a project called PREDICT that was run by the State Department, but it's been a big scientific enterprise across many institutions and many grants, and over many, many, many years, sort of a whole field devoted to understanding where are there viruses and other pathogens in animals that could spill over into humans. What is the probability that it's going to? What are the locations where it might be likely to happen? And what are the animal species that might harbor those viruses? And so this isn't as much a genetic enterprise as understanding the distribution of things in time and space, and understanding and characterizing risk. And so we have a different type of data we might be working with, not so much the genetic sequences, but we often have things like pairwise associations. So this is just a data table I pulled right out of a project of mine, where we have a big list of viruses and their associations with the types of animals that have been found to harbor them and the types of tests that have been used to collect that. And this is data that's been collected from decades and decades and decades of research that's represented in the literature, harvested from all that literature, and aggregated to say, what do we know about which viruses are in what animals, and what can we use to take that data and make predictions about where viruses might be in the future. So this is one type of data one might have when trying to do this sort of large scale risk type analysis. Another type of data one might have is spatial. So one might have an association of a virus with a species that it's hosted, but then you have to know where those viruses are and where those species are. So you might have records that look like this. There's a thing called the Global Biodiversity Information Facility. It's a huge global consensus database of records of where scientists have recorded the existence and the habitat of various species. So Rhinolophus affinis is the intermediate horseshoe bat. It's the one that the closest related sequence to coronavirus was found in Yunnan in China. And this is a map of records of places where it's been observed. So you can have this geospatial data, point data, saying here's a place where this species has been seen, here is a place where this species has been seen, here is a place where this species has been seen. And you understand data like this has its own problems and challenges for working with. And you're probably familiar with a bunch of these because it's a very common set of problems one has in a regression or supervised learning of various sorts. So one of the big problems we tend to have with this is sparse data. Unlike web traffic or credit card clicks or social media data, it's very expensive to collect a single data point often for this kind of thing. It's like to say a single row of data for me represents someone in the woods all night with a net to catch a rodent, draw its blood, swipe its butthole with a Q-tip, and let it go. And the fact that that happens thousands and thousands of times is pretty amazing, but you realize it's a very, very small sample of what's actually out there. And we know that sampling is highly biased. There are places that scientists work more. There are places that have been researched less. There are species that there's been more effort on. If you're trying to draw lots of data out of the scientific record or out of the collective databases, you know it's going to be biased in different ways. There are species that have been preferentially looked at for various reasons. Maybe they are of interest for reasons not related to disease. We know that collection happens more aggressively nearer population centers. That's where people observe things. You need an observer in order to find an animal. You have all sorts of correlation issues. You have geographic autocorrelation and correlation between types of species, that phylogenetic relationship. And then, you know, these type of predictions involve nonlinearity and interactions, and you have all of the challenge of dealing with prediction versus inference trade-offs. So you're trying to figure out where the viruses or the animals are. Are you trying to make predictions out of that for when you go back and do sampling in the future? Or are you trying to make a trade-off so you can understand some mechanism, which piece of, you know, what type of trait of an animal is most predictive or indicates the reason why an animal might have a virus in them? You want to understand that. And for that reason, all of these considerations, one throws a tool set that people who are general data scientists would probably be very familiar with at these types of problems. There are general linear models and generalized additive models, which are more on the inference side. You can have mechanistic models that very much explicitly model the type of habitat or the relationship between a host and its pathogen. One can use machine learning approach, not just maximum likelihood or machine learning approaches. Sometimes you're just interested in prediction. And so you can use non-parametric black box approaches to try to identify things in space. Or you might want to sort of use Bayesian approaches and be able to, once again, interrogate a posterior and do full Bayesian inference. And all of these considerations and these approaches are used for trying to do this general type of prediction. So I'm going to sort of give you a gallery of some of these. So this is a map from a paper put out by EcoHealth Alliance a few years ago, about which is sort of considered a risk map for places that may potentially have new viruses emerging. And it builds off a data set of historical new emergences of viruses into the human population from wildlife, and then tries to use that data for prediction. So this uses a boosted tree approach. The R packages involved with building it, or the Dismo package, which is a boosted tree approach specific to species distributions, but also XGBoost, which is a very general boosted regression tree approach. And the idea was to use a bunch of information about a location to understand whether it's a place that would be more or less at risk to having a new zoonotic disease spillover occur there. So you can understand that that's a regression problem or a supervised learning problem. And you have all of the challenges of, say, where there has been more or less surveillance. There are places where people have looked a lot and people have looked a little. So this was a time where we created a variable based on all of the locations that people have mentioned in the published literature related to disease. We took a natural language approach, tried to identify places that people studied more and studied less. And that highly biased sampling, we tried to address by then making a spatial layer that modified our model based on the degree of sampling and the degree of virus observation effort there had been over the decades in which these emergence events occurred. And then one wants to do not just potentially have predictions, but understand what's driving those predictions. And so for that, one might use partial dependence plots, which is a common way of interrogating a machine learning model. And so these are partial dependence plots of this boosted regression tree, but we wanted to understand uncertainty in them. So we created a bootstrapping approach that bootstrapped not just the data randomly, but the data according to that geographic bias. And that helped us understand the uncertainty around these partial effects and which variables made a place more or less likely to be a place where a new virus emerged out into the human population. So some of the variables that were important for this are the mammal biodiversity, the number of species that are out there that could potentially harbor viruses. There were several measures that are important related to tree cover. So general forested areas where people are encountering those animals are more likely. But it was also important that there were places where those forested areas run up against urban areas and highly settled areas, or even areas where there was land use change happening. People were clearing pasture. All those things made it more likely that a place would have an emerging disease occur over the course of decades, as this goes back to the 1940s. So that's, one approach we had looking at viruses and the geographic risk. Another thing that we're interested in looking at is what species are the ones that are hosts to different viruses. So in addition to where do viruses emerge, you want to ask the question and make the prediction, well, what types of species are more likely to be host to a virus that could eventually reach the human population? Once again, a big regression or supervised learning problem. Those of you who have seen me talk before at the meetup might know that sort of my favorite tool for this kind of thing is a GAM, or a generalized additive model, which is a nice sort of compromise between the more elaborate machine learning approaches, but also the more interpretable generalized linear model approaches. These are much like generalized linear models, but allow for nonlinear effects and let you sort of choose how they interact rather than automatically creating lots of crosses like in boosted trees. So once again, we had that problem of bias. Certain species are more heavily studied than others. And so we once again looked at, well, let's introduce a variable as to how many studies have been done on each species and use that as a measure of effort. And that's the biggest predictor of whether you find a virus in something. But one also finds effects of slightly bigger animals are more likely to have viruses in them. Ones that have big ranges, ones that have big ranges that overlap with other types of animals. So there's the potential for interaction between different species. And we were able to look at different categories of animals, different big taxonomic groups. So for instance, one thing that we find is that bats as a group, while there's a small effect, once you account for all of the other biases and everything else put in the model, overall are more likely to host some of these viruses than others. And that's something that's been seen in a lot of studies, though there was a lot of speculation as to why that might be and whether that has to do with sampling approaches or potentially has a lot to do with the physiology of the animals. Also, that last group on the right is, we also have primates and rodents and other ones that are potentially more hosts of more viruses that can spill over into humans. So this uses that type of regression approach. A colleague of mine, Greg Albury, who might be on here, has used this for another interesting approach of not just asking, well, which viruses spill from wildlife into humans, but from one wildlife species into another. Which viruses do we find in multiple groups? And given that we find a virus, say, in a bat species, what other species might we find it in? Because they're likely to share. And so we also use this generalized additive modeling approach because of sort of the ability to build these structured models with it, and found this combined relationship between the amount of space that species habitat ranges overlap with each other, and as well as their genetic relationship with each other, combine to help you predict a lot of the probability the virus can jump from one species to another species. And with that, you can sort of create this response service and ask, if you find a virus in one type of organism, what other types of organisms might it be in? And maybe those are interesting ones to go after for surveillance in order to understand, in order to potentially look for new potential spillovers and other animals that could potentially have viruses that spill over into people. And this work has also sort of extended into sort of a whole other sort of combination of models where some work led by a great researcher down at Georgetown, Colin Carlson, who may be here as well, has taken that along with these binary classification models for where species live in order to understand how climate change might change the distribution of viruses in the world. So one can create a model to understand where animal species live based on the climate. They prefer certain temperatures, they prefer certain seasonalities, they also prefer certain types of habitats. And one can create a binary classification or regression model with something like GLMNet. That's what was used here, with a lot of other bells and whistles in it too. And then take that and combine that with a model of, well, what predicts what viruses are in them and what viruses are shared. And then you can make projections or forecasts because you have a relationship between climate and habitat, as well as animals and viruses. And then you ask, how are the viruses going to move with their hosts as hosts move over time with climate change, as the climate in places change and the preferred habitat change. So all of these models that I look at, that sort of I've just talked about, things that help us understand essentially the in space and time, where do we see viruses and what species do we see them in and where we see those species, those are very much these regression machine learning approaches that may be familiar to you from all sorts of other data science work you do. And we encounter both problems that everyone encounters about data quality and sparsity. And then we have these things that are really unique to the problem of hosts and viruses and the way people observe them and the way that data is collected. So I'm going to turn to a third set, a very different set of models now for the last section, which is talk about how diseases actually spread in the population. The data, which I'll talk about in this, is something that you probably have all become quite familiar with following in the news. The data that we look at, that we try to understand things from and predict things from, are time series data, and time series data of things like the observation of new infections, new tested individuals, new hospitalized individuals, people who die from a disease. And we've all been following with sort of, great grim attention that for COVID over the past couple of months. What I'm showing here is a plot, I believe, of new cases from the coronavirus dashboard at Johns Hopkins University. This coronavirus.jhu.edu is a great resource for both visualization as well as raw data of cases of COVID that Johns Hopkins has developed. And this is the type of data one is dealing with. And it can have all sorts of challenges as well. So one gets time series data of these things, but there's all sorts of challenges with observation and the quality of the data. For instance, we know in many places there's not a consistent testing approach. So who is tested is might be not be the whole population or a random sample of the population. It gives you a different understanding of how many cases are actually out there. When hospitals report deaths or a nursing home reports deaths, sometimes it takes them a few days to report things. Sometimes it's slower to report things on busier days or over the weekends. So you get spikes on Monday. So you have all of these data quality challenges and difficulties in interpretation of these. And then you want to use this data to understand the progression of the disease, to maybe make forecasts about the disease, to try to take planning approaches to the disease. So you have a lot of considerations in terms of what you're doing. So maybe you're trying to forecast the future. You're trying to do some sort of time series in order to understand the capacity that's needed for healthcare. Or maybe you're not actually trying to forecast, but you're trying to scenario plan, because there's a lot of potential outcomes and a lot of different policy approaches that one might take. And you want to look at what the potential changes in the future might be if you look at those different scenarios, which may or may not be certain futures. You have the challenge that you're basically entirely doing out of sample predictions. We are sort of entering a new dynamic space that none of us has encountered before. When you're making a prediction, it's not a prediction that's trained off of lots of similar outbreaks. There have been no similar outbreaks. Perhaps you could say 1918 was a similar outbreak, but it was of a very different virus from a very different family. Lots of things were different. There are a lot of types of uncertainty that you might want to deal with or not want to deal with. So there's uncertainty in the parameters that you're putting into models that might be measured from the literature. There's uncertainty as to what the quality of the data is, and then often that data is partial. So you have all those considerations. And then similarly, you have a bunch of different approaches you might take to building a model of disease over time. So one can take a mechanistic approach that very much follows sort of principles of disease dynamics and epidemiology. One can take a phenomenological approach, which says, well, let's fit based on sort of previous events, and no matter sort of what drove that event, we're just going to sort of do a data-driven approach. One might think of things in a deterministic way, you know, A leads to B, and be primarily interested in, you know, the average effect, which is not that noisy. Or you might be interested in the noise and want to pursue the things that have to do with stochasticity and chance and your probability distributions. And so there's a lot of different models one might pick and different approaches one might take to what you're trying to do here, forecasting or planning, or trying to make some statistical inference about how the disease is working over time. So one type of model which has gotten a lot of attention and has been used a lot, both historically, epidemiologically, and very much now in the COVID-19 outbreak, is what's known as the SIR model, or the susceptible, infected, recovered model, which is a very classic, very old approach to modeling infectious diseases that has yielded, you know, enormous insights. And it's a very simple approach that can be made very, very complex. And I'm going to show an example of it that comes from a great Shiny app that you can go and look at, created by Allison Hill at Harvard Public Health. And she put together a Shiny app that demonstrates use of this model that's meant for sort of scenario planning. So these types of models break up the population into different groups of individuals: those that are susceptible to disease but haven't gotten it, those that are exposed but haven't necessarily been infected, those who have been infected, and then those could be any number of subclasses: those who have a severe case of the disease, those that have a mild case of the disease, those that have a case of the disease they don't even know is the disease because they don't have symptoms. And then both those individuals after they recover, and some of those individuals when they die. And each of those compartments is represented by a differential equation, which is a rate equation. It says how this group of people grows or shrinks depending on what's going on in all the other groups of people, or based on other external forcing factors. So that's all put together and represented as a set of differential equations, which is what I'm showing on the left. And when you use a differential equation solver in R or elsewhere to solve those equations, you get a simulation of what happens with those diseases going forward. So at Allison's site, you can do those types of simulations, which predict how many people will be infected, how many cases we'll have. Although I'll be careful to say it's not necessarily a prediction. So many of these models are not fit directly to data, but rather are highly dependent on parameters that might be measured elsewhere, or are choices that one makes. You know, it's very important that things like this often are represented with these sliders next to them, which is why they make great applications of things like Shiny apps, because there are values that we may or may not have good numbers for, and you can look at the scenario when they're high and the scenario when they're low and say, well, you know, what if we can get 10% of people who get the infection to self-isolate? Or what if we can get 90%? How does that change the dynamics of this system as representing these equations? You know, what happens if we totally reduce their infection rate when they're in the hospital, or we're not able to at all? All of those sliders, some of which represent things that are uncertainty, and some of which represent things that are choices, have huge effects on what the outcomes of these models are. And so they're often more about understanding the range of possible outcomes and the relationship between those inputs and the outputs as doing forecasting and prediction. And that's one of the reasons models are very different, and you hear about very different results, is that the assumptions going into them are different because they can often represent different policy scenarios or behavior scenarios. Now, the model that's shown here and the one that Allison showed is a deterministic model, but that's not necessarily what happens in the real world. There's a lot of chance in the real world. And so often what it wants to do is represent such models using stochastic processes. So much like one can say simulate from a linear regression using random sampling to represent the noise, a stochastic process, rather than simulating and then adding noise, has noise built into the simulation and built into the process. So given a set of assumptions or parameters at the beginning, you can have a very different set of outcomes because of those stochastic processes. And so this site, another really useful Shiny app, covidmeasures.github.io, is put together by a group in Stanford out of a lab of woman, Erin Mordecai, that similarly lets you sort of play with different intervention measures to understand what would happen in the case of different scenarios. But one of the things it can show you is the variance in those scenarios. So rather than just using differential equations to solve, it uses stochastic equations, which result in these many different possible outcomes, even given the same inputs. And that's important because the real world is noisy and random, and understanding the range of possibilities is just as important as understanding the average outcome. One plug I'll give for if you're interested in this space is an R package called Odin. So these differential equations, on one hand, you can have very small number of them and have a very simple model. But things get very complex when one starts thinking about space. So lots of different populations interacting from different places or over a network. Or things like demographics. You have lots of different people of different ages, each of which represents a group. And that can become very complex and computationally intense. Now, the best solvers are really available in R in a package called DESolve. And they're super fast because they're not written in R, they're written in Fortran. They go back to the 1970s and they've been maintained as sort of top of the line solvers ever since. But that means you have to write code in C or Fortran to make it run fast. This Odin package, which is put together by a guy named Ritzvich John, who is at Imperial College, actually part of that group that produced some of the famous early forecasts for COVID, has created a package that lets you write differential equations in R, and then it compiles that R code to C so it can run really fast. So it's kind of like a domain-specific language for these types of equations that you use to predict epidemiological processes. But sort of like dplyr, it has its own language, but when it executes, it does everything very quickly in C. So that's a great package. Rich is also part of the ARPANSci project, so this has gotten a lot of scrutiny, and it's something that's been used a lot for this type of work. Now, sometimes one is just simulating for parameters, but you can also take that time series data and fit to these mechanistic models, which is not as commonly done, but is certainly possible, and though it's something challenging. And that's when you, rather than just trying to fit a linear regression or an exponential curve, actually try to fit a whole dynamic model of this sort to observe data like deaths or hospitalizations or testing, understanding that's coming from uncertain data sources. In R, there are a lot of options for this. There's NLME, it's sort of the nonlinear fitting package, but also STAN has a full-blown differential equation solver inside it. So you can make models that have those dynamics and then fit data to it. Increasingly, the BRMS package, which lots of people who use for Bayesian model fitting, is putting together the facilities to do this as well. And if this is something you're interested in, there's a neat paper that shows sort of a comparison of different packages for doing it by Lee Dushoff and Ben Bulker, who some of you might know from creating the LME4 package that's really widely used, as well as a bunch of others in R. One thing that I really like to use is another variant of this, which is called a partially observed Markov process. So basically that idea is that we have stochasticity in our process and incomplete observations. And so you have two different types of uncertainty driving what one sees in the data. And so you sort of have this process model, which is this noisy, random series of events that occur underneath, and then sort of a noisy way of sampling that data on top of it. And that's the real world. We have randomness in what happens and imprecision in how we see it. So there's a great package for looking at those mechanistic models of that sort in R as well, called POMP, for partially observed Markov process. And so that will try to fit mechanistic models with uncertain parameters to this noisy type of data that you get in real life. And it's something I've been using a lot. And then parameters like the transmission rate or how long the course of the disease lasts can not only be drawn from other external sources, but be something that can be fit directly to the data. Aaron King's website has an absolutely fantastic series of tutorials and approaches to doing this. And it's a really interesting thing that one can use for epidemiological research and trying to fit models data to data to understand what sort of dynamics in the future disease will have, but also potentially lots of other real world nonlinear processes. So before I wrap up, I wanted to talk a little about things that our users and data scientists who are in this space can do to help COVID-19. There are lots of things we can all do besides staying at home, besides washing our hands, besides helping educate people and doing what you can, be it donating or supporting or helping take care of each other. I know lots of people who are data-focused people, who are quantitative people, sort of see everything that's out there and think they want to apply those skills. And there are ways that people can do that. And I think people who are attending this and are part of the meetup likely want to be able to help contribute in that way. So there are lots of ways and lots of organizations, but I'm going to talk just a little bit about one called RECON. RECON is the R Epidemics Consortium. It's been around for a number of years and has a focus on developing tools and training and sort of frontline deployment for helping epidemiologists gather and analyze data about frontline diseases. So it's a membership organization where lots of those members are epidemiologists trying to do work on the front line, from sort of forecasting future to just collecting and tidying and trying to report data in as fast and as helpful a way as possible. And so RECON has people that write software, that collect information, but then also actually sort of deploy to the front lines of epidemics. So they'll run training workshops, but a number of RECON members have gone to West Africa and to Central Africa to work on the Ebola outbreaks that have been there, to make sure that the data there is collected well, that it's tidied, that it's organized, that they can write automated reports that get delivered to policymakers so they can understand how those go. And many of the members there are working in sort of all of the countries that they are to support this type of work and develop sort of policy relevant data and models to medical workers and frontline policy workers and such as well. They have a really great website of resources where you can learn about those things. And then the core group develops a lot of R packages that are used by those epidemiologists. So these are ones that take in standard types of epidemiological data or help you organize that epidemiological data and make certain types of forecasts of hospital beds that are needed, trying to understand the current transmission rate in a place and time so you can see whether you're doing better or worse in terms of trying to control the disease. trying to organize information for contact tracing to go and find individuals who might have been exposed to the disease. There's a whole suite of R packages for people who do this work. And these types of R packages can use R developers to help fix bugs, to help develop. So recently, Recon has launched something called the COVID-19 Hub. And right now it sort of exists as a GitHub repository, but working with the R consortium, it's going to be built out into more of a broader interaction forum where people who are working in COVID-related epidemiology can post issues of problems that they have that they would like support from R programmers and data scientists and others to help solve. So for instance, sometimes a new source of data comes online, but it's not well organized and it needs to be scraped and turned into a package that collects that. Sometimes an epidemiologist develops a new tool, say a new way of estimating a value like to project hospital bed requirements. And they've done the math and they've done the initial version, but they could use help getting it pushed further along to be made into a package or made into a shiny app that people could consume. So the idea is COVID-19 Hub is a place that people who are working in those fields will be able to post those requests for the support, and people who are experienced data scientists and R software developers can pick those up and help them use it. I've posted things to this of, I found there's this new data source and it's going to take a bunch of time to figure out how to connect to it and pull it. And can someone set that up while we're working on other things that are high priority? And a lot of people have come through and there's been really great work. Sometimes all that's needed to do is just the coordination. You've put up a request and somebody else knows, oh, that thing is being worked on over there, you can connect. So people who are just sort of on top of things, even if you can't necessarily program, every type of problem that's there can really contribute. We are hoping with the R Consortium to get some dedicated time for someone to be a community manager to help connect people, because a lot of the challenge is, knowing the right people who have the skills that connect to certain problems. But this is a place that people can help with. So that's it. Thank you very much. It's an honor to be able to speak to such a large group. It's great to speak about an important topic. The New York R community and the New York R meetup has been really important to me, and an important part of the community in this city, and a community that continues to pull together to support each other in a really hard time, and support everyone else in the city and everyone else dealing with this work. So thank you very much, Jared, for having me. Thank you all for attending, and I'd be happy to answer any questions.
Thank you, Noam. So I compiled a few of the questions from people who are asking there. So let me pull them up. So the first question on my list is, in identifying the type of virus, have you researched any cure methods for them?
That's not work that I'm doing. There's a lot of work in both therapeutics and of course vaccines, which is sort of the most important, really the most important part of ultimately bringing an end to the pandemic. But one of the things that actually is important in this is that sort of early discovery of diversity of diseases and understanding those phylogenetic trees and relationships, because a lot of the work that has gone into developing potentially therapeutics or vaccines has gotten a head start from other SARS-like coronaviruses that have been found and have been tested, and people have thought about, if something like this comes along that we can look at, and if there's a new virus that's related to the current viruses, we can sort of get a head start on the types of therapies. So there's already been work going on in understanding possible cure, possible treatments, and possible ways of dealing with coronaviruses generally. There's a group down in South Carolina, the Barrick Lab, that has been working very hard on this for a long time, that collaborate with us because we often find some of the viruses, and they're the ones who try to understand which ones might be the worst and which ones might have some potential to help us understand future cures. So that's something that's related to that work, though it's not something that I've worked on myself.
Good. That was another question someone had. Can the phylogenetic modeling help us find a cure? So thank you for answering that one. Another question is, can you point to free data sets that they can use to explore this?
Well, there are a lot. I mean, it really is an amazing time with this. I will, when we follow up and post, and maybe after I wrap talking, I can post some in the Slack or the chat. But for instance, if you're interested in disease dynamics and spread and numbers, both that Johns Hopkins website, as well as the New York Times published, for the United States at least, publish really regular numbers in, the changes in cases and deaths and things like that at a very granular level. They've really been collect that stuff. There's also, if, I'm sure I'll get the ire of a lot of population geneticists of sending a bunch of people to, explore genetic data on their own, but genetic data is something that is super, super available because in science, it's been so standardized for so long, the formats. It's actually really a field that is very open, and that data is published with. So there's a site, GISAID, that is an international site where scientists post the sequences of the viruses that they get and they collect. That's what that Next Strain website builds off of. It gets all the sequences from there that it does that tree building. And then the Next Strain site itself is a great source because it points you to everything that it collects. And so you can see where the cases are and where the sequences are. And then some of the things, I'll post them, the websites of, where the data is of, say, the host and species relationship information. A lot of that is published alongside with our papers, and I'll put that in the notes as well.
Great. Another question we had is, what type of model are you using for geospatial data?
Well, there are lots of different ones. So if we're looking at, say, that what we call the hotspots model, which is the model, the risk map of where in space viruses are, that uses a boosted regression tree approach. I tend to use, because it's my hammer a lot, generalized additive models for geospatial data, because they're very good at incorporating autocorrelative geospatial effects. And if you're interested in that, I actually have an online course, I'll put a link to that too, which has a component on how to fit those models to geospatial data.
So is that built into MGCV?
Yes, that's the MGCV package.
All right, great. That package is magical. We can do just about everything. And for those of you who want to learn more about that too, I posted in the Slack channel links to Noam's video and his slides from when he spoke about this at the meetup a few years ago. And that's actually one of our top watched videos of all time. So it's a big hit. Another question we have is, what package did you use to produce the partial dependence plots?
Oh, so the partial dependence plots for those booster regression trees, that was definitely a very elaborate hand customized thing. So they're GG plots, but they were done from scratch, and not by me, by a great epidemiologist, Toth Allen, who is now at a place called the Recurse, works at the Recurse Center in New York. So he did a lot of that hotspot modeling work and is the lead on that paper. So that's what did that. There are a lot of good packages for that kind of thing. I like something called the Dalex package, which is for machine learning sort of model explanations. That's good for partial dependence plots when you have a sort of complex model like that. If you're interested in boosted regression trees, there's a new sort of Bayesian package, the Bayesian additive regression trees, and there's a neat package called Embarcadero, because they're BARTs, and BARTs is the San Francisco transit which stops at Embarcadero, and it's by Colin Carlson, and it automatically creates partial dependence plots for those tree-based models that it does. So those are great too. And
I am putting a link right now because you mentioned BARTs. I'm putting this in the Slack channel. Jennifer Hill gave a talk at the meetup a couple years ago about one of the topics she covered was BART. And the BART package, one of the BART packages was written by Vince Dory, who was there, and he should be speaking at the meetup at some point soon. Let's see. So you discussed about emergence from unintentional human interaction with wildlife. What about modeling about intentional interactions such as cultivation of wildlife for human use or consumption?
Yeah, I mean, that's an interesting, so, you know, it's a very common pattern that new diseases from wildlife actually pass through domesticated livestock before getting to humans, because domesticated livestock are often interacting with wildlife sort of at the edge of their range, but domesticated animals are often, say, in a much more crowded environment. So they're much more likely to be able to transmit the disease to each other and sort of cause an acceleration of disease transmission. And so that is something that has definitely been modeled in a lot of different ways. And so one is the degree to which those livestock are just like a predictor in like space and time of emergence. But it's often been a more mechanistic approach. So for instance, in Malaysia, and also in Bangladesh, there's a disease known as the Nipah virus that is transmitted from bats, but has infected humans through pigs. And so it's because the wildlife end up dropping like food scraps and feces and things like that into the place where the animals are harbored that you can get this spillover. So it's a common pattern. And there are a lot of different ways. So there's a paper we have that sort of shows how those pig dynamics actually are what drive the disease. It just needs to be seeded by the introduction from wildlife.
And you'll be able to post that to the Slack channel later when you're done?
Yep.
That'd be great. Another question I see here is, how similar are the different coronavirus viruses? And what are the chances that the wildlife hosts are tracking over time is misidentified due to convergent evolution?
I'm not 100% sure what they're asking, but I think I can answer it generally enough. And so there's a big diversity of coronaviruses, and they vary in a bunch of different ways. One of the major differences amongst the different viruses that's important is the part of their genome that encodes what's called a spike protein. And that's what's on the outside of the virus, and it's what helps it connect up with human cells and for the virus to be able to inject its DNA into those cells. So there's lots of differences amongst the different viruses. One of the things that's been found is that the pieces of the genome that encode that can recombine, because the different viruses, multiple viruses may be in the same host, and they can sort of swap parts. And the right swapping of parts together can produce the combination that is ideal for connecting up with another animal's or a human's type of cells using that spike protein. So you have a very large pool of viruses and these different parts amongst them, and sort of chance events let them recombine with each other. And there's a lot we don't know about this. You know, it can happen differently in the bat population versus other, you know, like domestic or other traded animals that perhaps the virus is in an intermediate stage before getting to humans. But it's really sort of this recombination, and like its sort of generality is one of the things that makes these different coronaviruses and the big pool of them a real big potential source for new viruses that can infect humans.
So that means you're saying there's a lot of them and probably a lot we haven't discovered?
Yes, there are definitely a lot we haven't discovered. That number sort of estimated is all over the place. You know, when you think about all viruses that could infect humans, there have been estimates that have come out that are in the millions. There are reasons to think that's much smaller because of sort of sampling bias approaches. There are reasons to think it's much bigger because viruses are constantly evolving and changing over time. But there's definitely a big pool of viruses that are unknown and potential other sources of infection, and doing things like being better at how we interact with wildlife, potentially, being able to do more early surveillance in human populations that interact with those wildlife, um, and sort of creating more of this sort of early warning system for those type of things, and understanding more of those viruses by sampling in the wild altogether can make it less likely that we'll be surprised again.
I apologize for that noise. I was getting Zoom bombed by a 1-year-old. He was having a lot of fun watching your talk. Yeah. So someone's asking a question about the different strains. And you already talked about the different strains, that we know there's different ones, but at what point do the different strains become different viruses? Can they mutate that far? Or could they only just become strains? And what's the difference between a strain and a whole new virus?
So it's a tough question in that, viruses more than other species, it's much harder to draw the line because they reproduce so differently. You don't sort of get this clear definition of, these viruses can interact with these and these can't. And so, the notion of a clade or a strain or a species is very fuzzy. But it's also important to know that you can have that mutation is a very normal part of viruses, even if they're not changing in any significant way. So those evolutionary trees, especially on something like next strain, which shows all of these different, all of this variation in those viruses, most of those differences are in genes or single nucleotides in genes that don't really have any effect on the virus. So like different groups of virus at this point, in our understanding, they might help us identify sort of like where a particular outbreak has come from, but there's not yet any evidence that those viruses are functionally different in any way. Coronaviruses like this, they like have that regular mutation. The big changes that have occurred in them seem to be partially due to that sort of recombination and swapping, which is more the case when you have multiple viruses in the same host. Sort of the real big question is not if the virus is going to change really in a big functional way, but how it might evolve in relation to an eventual vaccine. Because one of the things that we see in flu viruses is that when you have a vaccine, the flu viruses, that drives evolution in the flu viruses, and then the new viruses you have in the future are a little less responsive to that vaccine, which means you need to update the vaccine. And so it's a big open question as to how that will play out with the coronavirus. Previous coronaviruses, it's not been as big of an issue, but we don't know about this one yet.
I imagine that's something that's being studied to try to figure out how it will mutate, just like they study how the flu will mutate.
Right. I don't think we're anywhere near there yet in understanding that with this, as we don't have a vaccine or even a real good understanding of immunization and people's gain of immunity in reaction to it. But that's something that many people are very closely tracking in looking at the genetic sequences of the virus and how they will change over time once treatments are introduced.
And that actually just brought up a good point. I saw an article going around, and it's so hard to know what's true and what isn't just what's being posted on random people's pages. But have there been any vaccines for any coronaviruses before? Would this be the first time we've successfully done that?
Let me get this. So we have had, coronaviruses that have emerged, there has been the last SARS, something called Middle Eastern Respiratory Syndrome. And then there are a few widely circulating human coronaviruses that are largely associated with common colds. I do not believe there is an active vaccine for any of those. Both SARS and MERS have largely been controlled via other measures, though MERS continues to be an issue. And the other human coronaviruses haven't been as, they just like haven't been as damaging and haven't been as big of a problem. So sort of an active vaccine development hasn't been something for them. So I do not think there has been. That said, because of the understanding, because of MERS and SARS and the likelihood that there would be more coronaviruses, this is something, like for instance, that Barrick lab I mentioned, has started. So the there isn't a previous coronavirus vaccine, but most indications are that, you know, an active vaccine program will be able to produce one, and we hope it'll, you know, be produced soon.
And it's just, why is this coronavirus so much more damaging than the previous ones?
It's not something we totally know. It's something that there's, and, you know, sort of the molecular side of this is less my expertise. So I'm sure there's some people out there who will, you know, comment otherwise. But, you know, this virus is, it has a lower fatality rate than previous coronaviruses, but is much more transmissible. One of the things that maybe contributed to that transmissibility is its spike protein, that protein on the outside of the virus that binds to your cells, actually binds much more strongly. So it may be that that binding is part of it, and so it's more easily transmitted once it's in you, like it's more likely to actually take hold. It may be due to differences in transmission. If it survives a little better, you know, or a little differently outside the human host than the other ones, that could be part of it. You know, and all of those things combine, you know, it can be like a sort of a set of small marginal changes, a set of small marginal changes of how the immune system reacts to it and how it is able to defend itself against the immune system, that all add up. And then that top level, it's really about that super high transmissibility that results from those combined changes. And that's why it's been so much more damaging than other coronaviruses that we've seen.
There's been a few questions here about how can people contribute either through doing work or the data sets. And I believe you'll point us to the R Consortium site, but is that the best place for people to go to help both with time and data and anything else they can do?
I think so. I mean, that site's relatively new, so it's going to, we think it's going to expand a bunch, especially with the R Consortium coming in to help with the R Consortium to help with the R Epidemic Consortium. So I definitely point you to that first. Although one of the reasons I point you to that is that in my experience, the people who really like need a little bit of support, they're like the big groups doing, you know, like sort of high level modeling. But often, you know, if you live in a community where, you know, people are thinking about, you know, where there are first responders and like one hospital and, you know, small groups, often the capacity just for managing data and handling data and doing that kind of thing is not as big as there is in, say, like the New York Department of Public Health that's doing a lot of planning. And it's been my experience that a lot of requests from help have come from small groups like that kind of flowing around, and someone who can just, you know, organize spreadsheets and make graphs and keep them up is often helpful. But it's hard to connect with those, right? You have to sort of have that connection with your community. And so if you do have that type of connection, I'd encourage you to find it. Otherwise, the best thing that I know is the R Consortium for now, but I think something we will try to do is post more links and connections to other groups to make sure that we're pointing people to other resources.
And please, people, do make use of the COVID-19 channel, post in there and get help in there and offer help in there too, because this is a big community of data scientists and epidemiologists. And I'm sure there's a lot of them amongst us besides just Noam. Another question was, how do you analyze DNA sequences? Do you tokenize the sequences and analyze relationships?
So that's a big question, and I have to emphasize, like most of my work is in that big geospatial stuff and the dynamic stuff, and there are many ways to analyze those DNA sequences. There are contexts in which tokenization is used. You know, there are like some big sort of machine learning approaches, especially when you're working with full genomes and trying to do prediction off of those. People will do sort of clustering and tokenization algorithms in order to reduce those down and use them in regressions. But when you're doing the sort of stuff like the phylogenetics that builds off the evolutionary sort of structures to build the model and those processes, you really are handling those at the individual nucleotide level. There's of that one letter in the string, or short groups of those that represent single proteins or single genes. And so it really is sort of specialized. And then when it comes to being like, well, let's take a genetic sequence and do a big machine learning approach to do prediction, then that often does fall into many things that are more familiar to broader scientists. There is work that's been done essentially taking an entire genome sequences and doing giant logistic regression on every nucleotide. It's not always, some of that is sort of hand wavy work that isn't as useful, but some of it's good. And so there, a lot of the techniques that are used to reduce those models, which otherwise would be overly large with overly wide data. People have used like Val-Pal-Wabbit, which is I forget, is either Microsoft or a Facebook project.
John Langford.
Yeah. To do like massive, mostly logistic regression, people have thrown that at the genetic data, or things like, GLMNet at genetic data too, essentially are trying to deal with many, turning those sequences into many, many variables. That, or you're going into that really sort of genomic specific software and models.
And GLMNet was actually invented for genomics, actually. That's Hasty Tripshiani did it specifically for that. And I just posted a link to the Slack channel for John Langford's talk about Val-Pal-Wabbit about a year ago at the meetup. So if people want to find that, that's over there. I see that Michael just posted the actual presentation itself to the chat over here. And Michael, that's not up on the meetup website, the actual PDF, so you could put that up there. That'd be really awesome too. Let me see if there's any other questions here. Someone's asking, what are some good resources to learn about epidemiology and public health, like books, websites? What other resources do you have besides your excellent talk? Where can they go learn more?
Oh man, that's big. I'm not gonna have one of the things about epidemiology is it's such a big field. You sort of remember, we have this focus, especially now, on infectious disease epidemiologists and, these particular types of models and particular type of focus. But epidemiology is the study, of health in populations, and disease ecology of health in populations of wild animals and plants. So some epidemiologists are thinking about, air pollution burdens on people, and some are thinking about cancers, and some are thinking about transmissible stuff like influenzas and COVID and AIDS. So it's a very big topic. So to some degree, sort of diving in to something more specific is often the case. So I could probably post some good ideas about where to learn things about, like, these infectious disease dynamical ODE models that people could look at, and some of the, not just the models, but the underlying principles and theories. And I'll come up with some and add them. But when it comes to epidemiology, it's as big a field as anything else. Trying to learn about biology or learn about physics, it's often very helpful to figure out a smaller piece to chew.
Yeah, there's a lot there. So maybe you just post, some of your favorites, like, the landmarks, like elements of the equivalent of elements of statistical learning. If you think of one, post it to the Slack channel.
Right.
Dan Chen, I'm specifically calling out his name because he does bioinformatics, asked, with all these resources, where's the point of no return where you have too many and therefore data overloaded and can't make meaning out of it all?
We are simultaneously way past that and not far enough, in the sense that, like, there is lots and lots of, every day there's more data on COVID, and there's lots of this. On the other hand, there's still a lot of data we don't have. Like, one of the big things that's hopefully coming online in the next few weeks is people doing antibody tests, and there being data coming out from antibody tests of people that helps us understand who has previously gotten the disease. So who might have gotten the disease that haven't had symptoms, or have had flu-like symptoms, but don't know if it was COVID or not, and sort of let it pass without being tested themselves for the live COVID. So, that type of information is going to be, useful. At the same time, then there's just this flood of stuff. If you follow the scientific literature, it's been fantastically, rapid and fast. And in terms of the rate at which we've learned about this disease and the rate at which we are reporting about it, it's like nothing else that, has happened in any other epidemic. Even people who spend all of their time thinking about this can't keep up with it. So what's really helpful is often the people now who are writing the reviews and the summaries and the aggregates of things. I've come to rely on those a lot.
Speaking of which, we just had a question. What would you say to people who aren't experts in the field, but they're trying to play the data to make sense of it and model the future spread of the virus? How can they make definitive statements about how it will progress?
I mean, I don't think we can never make definitive statements. You know, we can tend to make statements of more and less probability. And, you know, I'm not one that thinks that, you know, like only an epidemiologist should run such a model, or that only certain types of science do certain types of work. But I think, you know, if you're trying to play with the data and learn with it, and you want to help others learn and gain insight with it, the best thing to do is to try to engage with others who have different types of expertise in the field, right? So people who are not, you know, whose main expertise was not in epidemiology or certain types of medical expertise, have made sort of great contributions to the science, but it's best done really in dialogue. So I'd say if you've like pulled something up and played with it, and you have like what you think is a really interesting insight, it's a really good idea to see if you can find someone to like bounce that off of, who has direct field expertise, or at least has worked in the related area, because you'll have your own biases from your areas of expertise in your fields, and they'll have different ones, and people can help people, you know, make catch problems that come up, or they know something about the origin of the data that might not be immediate obvious, or they might know something about principles in the field that this either supports or clashes with, and you have to figure out why, or if something needs to change in your work or the principles. And so I think it's great for people to work with this data, play with this data, share with each other the type of work. But if you think you like have something that you want to make a big statement about because you found something, it's something that you really should make an effort to collaborate first, because that sort of helps cut down the noise for a lot of people, and helps people find mistakes before they can cause a problem or result in some misinterpretation or misinformation.
I think that's a very good point. Rob Heinemann, who is a world famous time series and forecasting expert, put out a blog post a week or two ago saying you can't just use like regular time series models for this, univariate time series models, even multivariate, because it's so much more than just a time series model. Time series is great when we don't know underlying processes, but fortunately, a lot of epidemiology, there's other information we can put in there, and you just don't want to look at deaths by day or infections by day, because you're missing out on so much more. So really think about the methods, and don't just go for like, hey, it's over time, let's look at time series. You got to think about spatial, got to think about all the epi stuff. So really collaborate, as Noam was saying. We have more questions coming in about how can they contribute with data. And again, you've mentioned that our consortium, that COVID resources site, which later, if you could post a link to the Slack channel, it'd be great for everyone to have that.
So why don't you pick one more question, Jared?
All right, got it. So this is actually a follow-up from what you said earlier about the phylogenetic modeling and about which drugs might work against COVID. Like for instance, if COVID's in the same cluster as drugs for which HCQ has worked before, would that give good evidence that HCQ would work again?
This is probably the furthest from my expertise of any question we've had.
All right, let me ask you a different one then.
I'll give it a short. So the short answer is I really don't know on that front, but to the extent it's like a way that people help identify candidates, it does happen, but it tends to happen much further upstream. The phylogenetics help you sort of think about whether do tests about whether the viruses work the same way, and then the tests sort of come later.
Great.
But I couldn't tell you more than that.
Fair enough.
You can do one more, since I couldn't really answer that one.
Fair enough. Another one's just asking about why is transmission so high in the northeast of the United States? And yeah, let's just go with that. Is it demonstrably higher in the northeast than other places? And is there a good reason besides density in New York City?
Well, there's a lot of theories about this. It's the kind of thing you think you can have very intuitive reasons, and it's tough to figure out why. People think about density and the different nature of the cities, you know, and but another important thing can be timing of policies. You know, to our understanding, West Coast cities were able to enact, you know, shelter in place and, you know, pause and social distancing policies earlier in the epidemic than elsewhere. And so that's one possibility. You know, density is another possibility as well. One early possibility is maybe that a lot more people with the disease were showing up earlier, and that actually drove early growth more than we knew. So these are all possibilities. It's something that we're looking at a fair bit now, because it's very important in understanding the approaches to managing disease in the future. But the density question is one that's still out, is still out to be answered. There's some evidence that that type of thing has made differences in other diseases in the past, but not necessarily as much as one might think.
So I want to say thank you very much to Noam. I hope we all have a certain measure of comfort in our lives seeing that people like Noam are working on this and really putting in the best effort to solve this problem and hopefully get us back to normalcy. Normally at this point I tell everyone to give a big round of applause for you, but you won't hear it. So just everyone imagine, I hope we're all applauding Noam at home individually, and is nice and raucous, and very thankful for that. So everyone else, you can absolutely go ask questions in the Slack channel. Maybe later tomorrow, Noam's not so tired, he'll be able to hop in there a little bit, use the COVID-19 channel as other resources, and you know, stay tuned for next month's meetup. We will continue and have another talk about coronavirus, because odds are good we might still be back in our homes. It'll be nice if we're not. So in the meantime, just thank you very much, Noam. I hope everyone has a wonderful night or evening or day or afternoon or morning, wherever you are, and see you around virtually in our community. Have a great rest of your time. Thank you, Noam.
Thank you, Jared, and thank you, everybody.
I see a lot of people clapping in the comments for you, so great job, Noam.